Okay, folks, this is our fourth video, but it's the third PowerPoint for this chapter. I broke the last one into two different videos, but this is part C or part three of digestive system. Let's jump in. Okay, so we're going to talk about the small intestine now. Small intestine is the major organ of digestion. It is not the stomach. You need to remember that. That's important. The small intestine, we've got three parts. We have the duodenum, the jejunum, and the ileum. The first part by the stomach is the duodenum. That is everything that is past the pyloric sphincter on the stomach. And the jejunum is the middle section. It's very sausage coily looking, because that's actually what sausage coils are made out of, using pig jejunum. And then the ileum would be the last section of the small intestine, and it ends at the ileocecal valve. Once you go through that, you're in the large intestine. Now, it's important that you understand large intestine, small intestine, those are terms describing the diameter, not the length. The small intestine is actually longer than the large intestine. All right, so the small intestine is 2 to 4 meters long from the pyloric sphincter to the ileocecal valve, so valve to valve. And it's about 2.5 to 4 centimeters in diameter. So 1 inch or more smaller than 2 inches. That would be 5 centimeters. Anyway, the parts, the duodenum, the jejunum, the ileum. So the first 10 inches that are wrapped around the head of the pancreas, that's where we do most of the reabsorption. And that is actually mostly embedded in the wall behind the peritoneum. So the duodenum, unlike the others, is mostly retroperitoneal. The jejunum, like I said, kind of sausage coil looking. It's attached at the front by a mesentery. It's about 8 feet long. The ileum is attached to the back of the body by mesentery. That's what goes to the ileocecal valve and then hooks up to the large intestine. That's about 12 feet long. All right, so let's move on. Here we have our parts. They're color coded. You can see that this small part right here in purple, that's the duodenum. The much longer in blue, this is your jejunum. And the much, much longer here in purple, this is your ileum. We end at the ileocecal valve, and the cecum is, in us, this little vermiform appendix. In other animals, it can be as big as the stomach or even bigger than the stomach. All right, moving on. So we look here at this picture that we've seen before. We have a liver, we have a duodenum. The right here at the opening to the duodenum, we would be going through the pyloric sphincter off the stomach. As that acidic chyme comes out of the stomach and travels down here, we are going to empty in at this major duodenal papilla. We're going to empty in basic fluid from the pancreas to neutralize it. We're going to empty in bile from the gallbladder to emulsify fats so they can be digested. So blood to the small intestine comes through. Are we talking small intestine or are we talking entire? Yeah, we're talking entire small intestine. Blood supply comes through the mesenteric artery, and there are veins that are carrying nutrient-rich blood from the digestive system, and they carry that to the superior mesenteric veins, which then go into the hepatic portal vein, which then go into the liver, where it's processed and detoxified. Parasympathetic nerves from the parasympathetic signals from the vagus nerve sympathetic from the splanchnic nerve. Those control what we're doing with our duodenum, meaning it's autonomic nervous system. All right, modifications of small intestine for absorption. Surface area, surface area, surface area. What we're talking about here is we need to have lots of surface so we have lots of area to do absorption. What we're talking about, there are ridges. You can actually see the gross anatomy ones in this drawing. You see those kind of ridges or circles there? Those are called circular folds. And the circular folds go around, they're just more surface area. On the circular folds, we have villi that stick up, finger-like projections that stick up that increase surface area. And then on each one of those villi that stick out, those villus that stick up. So here we have villi. And imagine my head is the top of one of those villi, but here we have a whole bunch of villi. Then you have microvilli covering all of that. So on the wall of the duodenum, you have circular folds, and on the circular folds, you have the villi, and on the villi, you have the microvilli. The microvilli are so small, they form what we call the brush border. So circular folds are about 1 centimeter deep. They make the chyme spiral as it goes through the intestine. In some animals, they're super well developed. In humans, they're there. If it takes more time to pass through the intestine, then you take more time to get energy out. The finger-like projections of mucosa about 1 millimeter high are called the villi. They have a capillary bed in the middle and a lot of lymphatics capillaries to help them absorb. Remember, those are lacteals. The capillaries absorb fat. The microvilli are cytoplasmic extensions of the cell membrane. They give a fuzzy appearance that we call the brush border. And that brush border actually has on it special enzymes for breaking down nutrients. They're called brush border enzymes. Super good. There are brush border enzymes for carbs, brush border enzymes for protein. They're both really good at doing their job. All right. So that's a nice drawing. You can see the circular folds, and you can see that these circular folds have villi on them, and the villi have microvilli. Good, good. Now, these villi, let's see, structural modification, blah, blah, blah. Yeah, you can see the capillary bed inside of, for both blood and lymph, inside this cutaway of a villus. And there's pictures of the same thing in real life. All right, let's move on. They're the microvilli, the brush borders. Okay, those are the best thing for absorption. You have enzymes that are embedded into the brush border. So modification of the mucosa and submucosa of the small intestine reflect function. They're intestinal crypts, spaces. Those are tubular glands between the villi. There are 5 main types of cells in the villi and crypts. There are enterocytes. That's most of the epithelium. That literally means inside cells. It's simple columnar absorptive cells, tight junctions, and mini microvilli. Great. Their job is to absorb nutrients, absorb electrolytes. The crypts, the spaces, are producing intestinal juice. This is watery mucus that's a carrier for the chyme. We also have in the wall goblet cells, mucus secreting cells. found in the epithelia and in the villi and crypts. Those are probably going to lubricate and make it easy to move the chyme along. We have enteroendocrine cells. That's where enterogastrins are made. And these are scattered in the villi, but some are down in the crypts. Let's see, plant cells found deep in crypts. Special secretory cells that fortify intestinal defenses. Yeah. Let's see. And you have stem cells that continuously make other types of cells. Yeah. All right. Mucosa associated lymphatic tissue, remember MALT tissue, protects against microorganisms they're trying to invade. This could be lymphoid follicles. They're like ultra mini tiny lymph nodes, but they don't have the connective tissue around them necessarily. Or Peyer's patches, which are even more like lymph nodes. Those are in the lamina propria. We have a ton of them in the near the end of the small intestine. Why? Well, that's where our bacterial numbers go up. So it's probably a good place to have a sentry system looking for bad bacteria. The lamina propria also has a whole bunch of plasma cells that make, remember, plasma cells are grown up lymphocytes, B lymphocytes that form, that they basically become an antibody factory. So we have lots of plasma cells there making immunoglobulin A. The submucosa has lots of areolar connective tissue, and it has in it duodenal glands of duodenum secrete alkaline mucus to neutralize the acidic chyme. Great. Okay. Not like we haven't mentioned that before. That method, it works, but we're doing it here. All right. In chemotherapy, this targets cells that divide rapidly. We're talking cancer cells. But there's a lot of collateral damage. It hits other rapidly growing cells, and that includes the epithelium of the GI tract. If you wonder why cancer patients on chemo get nauseated so suddenly, those cells are collateral damage. The lining of your GI tract, those cells are going to get damaged. It is what it is. All right. I'm going to put this on pause for a second. I'll be right back. Hold on. Okay, so what I was saying, chemo goes for the fast dividing cells because cancer cells are really fast. So when you take chemo, you're trying to kill the fast growing cells, hoping it'll be mostly cancer cells. But there will be other cells that get attacked. And because the lining to your stomach is reproducing so fast, lining to your GI tract, that's going to get attacked too. A lot of those cells are going to die. That's going to cause nausea, vomiting, diarrhea, chemical side effects that we often hear about for chemo. And you may have wondered why in the past. Well, now you know. All right. So intestinal juice, you're going to make a liter or two of that every day in response to the intestines swelling as they're filled with food or irritation of the mucosa by the acid of the chyme. The major stimulus, what did I say chyme? It's chyme. The major stimulus for production is hypertonic or acidic chyme. The fluid that you're making is slightly alkaline. It's isotonic with blood plasma, so it will be able to dilute any of the hypertonic conditions. It's mostly water, but it also has some mucus in it, mucus from the duodenal glands and the goblet cells of the mucosa. The chyme that comes in from the stomach is going to have partially digested carbohydrates, proteins, undigested fats. It'll take 3 to 6 hours in the small intestine to absorb all the nutrients and most of the water. And then it'll go on to the large intestine where you absorb even more of the water and the B vitamins. Anyway, So sources of enzyme. Where does the enzyme come from that we use for digestion in the small intestine? Bile, bicarbonate, digestive enzymes from the liver and the pancreas, brush border enzymes that are found in the villi and microvilli in the epithelial that lines the lumen. You notice stomach is not on the list, and saliva is not on the list. Why is that? Well, the salivary enzymes, when they hit the stomach, they're denatured by the strong acidity. The stomach enzymes, they're released in the stomach. They work well. Their optimum is in that high acidity. When they hit the intestines, the duodenum, and we dump the basic pancreatic fluid in, those stomach enzymes are going to denature. So the enzymes are still there, but they're not usable. And that's why we have new sets of enzymes for the intestine. Those are ones that work good at the pH we're trying to keep the intestine at. All right, moving on. So chyme coming into the duodenum is usually hypertonic. It's concentrated. So chyme delivery has to be slow. If you did it fast, water wants to dilute stuff. Water would be leaving the blood and going into the intestines. We don't want that to happen, so we do it really slow. The chyme also has a low pH. I already told you that that'll destroy the intestines, so we have to adjust the pH upward. We do that by mixing it with pancreatic juice. We also mix it with bile to emulsify the fats. And enterogastric reflex, enterogastrones, the hormones, they control the movement of food into the duodenum to prevent it from being overwhelmed. It's sensitive and it doesn't have all the defensive structures the stomach does. All right, so motility. After a meal, segmentation is the most common motion of the small intestine. You squeeze and push the things apart and squeeze and push them back together. All right. This is initiated by intrinsic pacemaker cells. We have pacemaker cells that control the muscle movement. This mixes and moves the contents towards the ileocecal valve. That would be more of the peristalsis happening. And the intensity of the contractions is altered by extrinsic nerve signals from long and short reflexes that are those are gut brain ones. Those are short distance. Also hormones. The parasympathetic will increase motility. What that means is the parasympathetic, when we're not under stress, we're not under fight or flight, it's going to increase the digestion, the movement of things through the digestive tract. When we're having to run from a bear, we're not going to be worrying about moving the food through the intestine. All right, so between meals, the amount of peristalsis increases. And we even have a hormone with a great name. Motilin causes motility, causes movement. So initiated by the motilin in the late intestinal phase every 90 to 120 minutes. Each wave will start distal to the previous wave and return to the migrating motor complex. Meal remnants, bacteria, debris, we're moving it all towards the large intestine. The complete trip through the small intestine from duodenum to the end of the ileum is probably taking about 2 hours. All right. This is, you've already seen this before, the peristalsis and segmentation. This is from the very first PowerPoint on this chapter. All right, so the ileocecal valve, this is the valve at the end of the ileum. It controls the flow of food into the large intestine, going by the cecum, which we in humans call the appendix. So the ileocecal sphincter will relax and let chyme into the large intestine. Under what conditions? Well, when the reflex is enhanced by segmentation in the ileum, the gastrum will increase motility in the ileum. The ileocecal valve flaps will close when chyme exerts back pressure, and that prevents regurgitation into the ileum. Fancy way of saying, if we are passing food through and it goes through the valve, when the intestine tries to push it back, the valve stays shut. Keeps stuff that moved from small to large in the large. All right, so table 23.3. Yeah, this is a long table. Okay. Table 23.3. Control of small intestine motility. So what does the stomach tell it to do? What does stretching of the small intestine tell it to do? What happens in the mouth? Yeah. Skim this. I'm not saying I won't ask things that are on this table, but I am not specifically trying to. I'm going to be pulling questions from the slides. So 23.3, that table is worth looking at to review yourself. Okay. Now let's talk the large intestine. Okay, the large intestine, remember, is large diameter. It's actually shorter length than the small intestine. So the large intestine says 3 unique features not seen elsewhere. The tennae coli, 3 bands of longitudinal smooth muscle in the muscularis. Let me reword that and say it differently in a way that'll make more sense. The muscularis mucosa is so reduced that you've lost the circular layer, and the longitudinal layer is so reduced it's not a continuous all the way around layer anymore. Instead, you have 3 little ribbons of muscle on it, and those are enough to hold some tension and create pockets or sacs that are called haustra. They're caused by the tension of the tone in the tennae coli. You also have epiploic appendages. It's a fancy name for little fat-filled pouches of visceral peritoneum. Remember visceral and parietal, and the visceral is stuck to the organ? Well, imagine that peritoneum, you have a bulge in it, and that bulge is full of fat. All right, so subdivisions of the large intestine. Before we do that, let's do subdivisions of the small to refresh our memory. The small intestine is divided into the duodenum, the jejunum, and the ileum. All right, the large intestine is going to be divided into the cecum, that's the appendix. And then in humans who have a simpler system than, say, a horse, you're going to have an ascending colon, a transverse colon, a descending colon, and a sigmoid colon. The ascending is on the right side of the body. The transverse is across the top near the diaphragm. The descending is on the left side, and the sigmoid is from the left to the middle. All right, so the and let's see, do they, yeah, they have it there. They should have put them, I think, in the, we'll go through and then we'll come back to the slide. So the large intestine has the cecum, the appendix, and then the ascending colon, transverse colon, descending colon, sigmoid colon. Okay. And then they're including as large intestine. I've seen people break them out differently, but they're including then the rectum and the anus as a part of the large intestine. Anyway, so let's back up and jump through these. The cecum is the first part of the large intestine. In like a rabbit, a horse, that's a huge thing. Any of the hindgut fermenting plant eating animals, that's going to be how they do it. In us, it's greatly reduced, but it's not like useless or anything. We do have mucosal associated lymphatic tissue there. We have bacteria stored inside, and we have lymphocytes stored inside. If we wipe out all the microbes living in the gut, have a disease or something, or take a broad spectrum antibiotic and we kill most of our microbiome, we can repopulate it from this. The twisted shape of the appendix makes it easy to get blockages in, which is not good. Now, the colon, large intestine, we have the ascending, the transverse, the descending, the sigmoid. You heard that already. Most of them, the transverse and the sigmoid, are in the peritoneal cavity, but the ascending and the descending, they're kind of at least partially retroperitoneal. So the ascending colon travels up the right side of the abdominal cavity to the level of the right kidney. You make a right angle turn. It has a name. We call it the right colic flexure. We go across the transverse colon to the left side. Then we have the left colic or splenic flexure. And then we have the descending colon that goes down the left side of the abdominal cavity. It doesn't go quite all the way down because then you have a sigmoid colon, the S-shaped portion, that comes back to the middle, from the left to the middle, and goes down through the pelvis. Then we have the rectum. Rectum is based on the word rectus, which means straight. You might remember the straight muscles down the middle of your stomach are the rectus abdominis, or the straight muscles is part of your quadriceps over your femur, or the rectus femoris. Rectus just means straight. So the rectum is straighter than the other pieces of large intestine. Anyway, there are 3 valves to stop feces from being passed along with gas. That works most of the time. The anal canal is the last part of the large intestine. It opens to the body exterior at the anus. There are 2 anal sphincters. The inner one is smooth muscle. The outer one is skeletal muscle. And there we have a picture. Okay, great. So these little pouches filled with fat, those are going to be epiploic bodies. Oh, there they say it, epiploic appendages. And here we have the appendix, the cecum, the bottom there. Then we have ascending colon, transverse colon, descending colon, sigmoid colon, and rectum. Okay, great. Other things to mention. I'm not seeing, here it is. Hostra, these are the pouches that we were talking about. Those exist because the tennae coli, what's left of the muscularis externa, you see one of these here, those are pulling on the intestine, keeping it a little bit shorter than it could be. If you had a piece of intestine that could be that long, and you pull on it, it's going to form little pouches. Those are the hostra. Okay. So inside of the rectum here, we have the rectal valve, we have the rectum, hemorrhoidal veins. These are just veins for that area. They have the name hemorrhoidal veins because those are what will become hemorrhoids if they turn into varicose veins. The anal canal is where we pass the feces through. The external anal sphincter is these muscles right here. The internal sphincter obviously is internal. These are muscles here. Both of those are going to squeeze and hold the anus shut. We'll talk more about that later. We have anal sinuses here. Sinus meaning space. There is not a lot of people think, well, shouldn't we have lubrication there, mucus? We really don't have that much of that going on. Anyway, so relationship of large intestine to the peritoneum. The appendix, the cecum, and the rectum are retroperitoneal. They're behind the peritoneal cavity. The colon is also retroperitoneal, except for the transverse and sigmoid parts. The intraperitoneal parts, that would be the transverse, the sigmoid, are anchored to the posterior abdominal wall by the mesenteric sheaths we call mesocolon. Meso meaning middle. Okay, so there we have it. Remember, the gradient omentum is like a curtain of membrane full of fat that they thought looked kind of like lace. You really have to use your imagination to get lace. But you can see a curtain of membranes with lots of fat globules in it. That's easy. All right. So lesser omentum goes from lesser curve of stomach up to liver. Greater omentum they have cut off here, but it would go from the greater curvature of the stomach and hang down over the intestines. We've been through everything else on this slide. All right, greater omentum lifted up, and then you can see, looking at this, the transverse colon is here, descending there, sigmoid there. What they've done is pulled the jejunum and ileum out of the way. That's why you don't see the ascending colon coming up there. This is pulled out of the way so it can show the mesocolon, so it can show the mesenteric arteries, the mesentery. If you pull intestines out of a dissected pig, you're going to have this mesentery attached to the intestines. So as you take the intestines out, there'll be these sheets of mesentery that are full of blood vessels, full of lymphatic vessels, full of nerves. All right, here we have a view from the side. You can see, if this is our stomach right here, we have the lesser omentum there. The greater omentum is this big loop that's hanging down and covering most of the intestines. The duodenum is retroperitoneal. The jejunum and ileum are not. Those are in the middle. Let's see. Transverse colon is in the middle. Yeah, okay, that's straightforward. And let's move down. So, okay, appendicitis. The appendix we talked about is that little worm-like extension off of the cecum. You could have it get inflamed. It could happen because it gets blocked by feces going by. This is most common in adolescence because the appendix is widest at the opening in adolescence. You could make it hard to drain venous, for the venous system to drain. That could cause ischemia, your no blood delivery. You could have necrosis, tissue death. Ruptured appendix would cause peritonitis. So symptoms: pain in the umbilical region, moving to the lower right abdominal quadrant, loss of appetite, nausea, vomiting. They can surgically remove the appendix to treat this. That's called an appendectomy. But in some cases, you can just do it with antibiotics. So the large intestine has thicker mucosa, and remember, that makes sense. Most of our absorption is in the small intestine. So the small intestine has a very thin wall to make it easy to pass the nutrients across. The larger intestine, that mucosa, that wall is thicker, and it is simple columnar. This is not true when you get to the anal canal. Why? Because along the length of the large intestine, we're absorbing water, absorbing water, absorbing water, concentrating the feces, packing it close together. When we actually get down to the anal canal, we have a lot of stuff rubbing on the wall. So we don't want to simple anything. We want to stratify layers. So we're stratified squamous, the same as we are anywhere that we have a need for abrasion resistance. All right. The large intestine mucosa doesn't have circular folds, doesn't have villi or microvilli, doesn't have digestive secretions. It does have a lot of crypts with mucus-producing goblet cells. Why? Because as we concentrate the feces, it'd be nice to mix some mucus in there so they can slide along and easily, coat them in mucus. Um, the mucosa of the anal canal will hang in long folds or ridges that we call anal columns. Anal recesses located between the columns. They do secrete some mucus to help in emptying, but you don't do, it's not serious lubricant. The pectinate line is a horizontal line parallel to the anal sinuses. There are visceral sensory nerves there. That region doesn't feel pain. The somatic nerves that are inferior to that line, they do feel pain. Superficial venous plexuses of the anal canal will form hemorrhoids if they become inflamed. Here's everything we were just talking about. It's the same picture you saw before. And bacterial flora, more than 1,000 types of bacteria. They outnumber our own cells 10 to 1. They enter from the small intestine or the anus. They can come either way to colonize the colon. They do have important functions, metabolic functions. Fermentation, they can break down carbohydrates that we couldn't digest, break down mucin. But when they do that, they're releasing irritating acids or noxious gases. It doesn't say this here, but often filled with sulfur compounds, around a half a liter a day, 500 milliliters a day. I think unless you're my dog, then it's more. Just guessing. These bacteria also make vitamin B and some vitamin K, so we'll absorb them there. Keeping pathogenic bacteria in check. Beneficial bacteria outnumber and suppress the bad ones. The immune system will destroy any bacteria, good or bad, that tries to come through the mucosa. The epithelial cells will recruit dendritic cells. Remember, those are antigen-presenting cells to the mucosa, and those antigen-presenting cells, those dendritic cells, will look at microbial antigens, and there are T cells living in the malt, the mucosa-associated lymphatic tissue. Well, those dendritic cells will show the T cells anything that looks suspicious, and that will trigger antibodies, immunoglobulins, to be released that will restrict the microbes and keep them in check. Gut bacteria and health. So we have a lot of mounting evidence supports the findings that gut bacteria can influence your body weight, how susceptible you are to different diseases, our moods. Yeah, like nerves, our nervous system affected by what bacteria we have living in our gut. Manipulating gut bacteria may become a routine healthcare strategy in the future. It's experimental and kind of creepy, but we already have situations where they do fecal transplants with unhealthy people to give them a healthy microbiome. Usually this is for really critical things, not for like weight loss, but it's important to know. Okay, so homeostatic imbalance. Let's talk about antibiotic associated diarrhea. That accounts for 14,000 deaths a year. Clostridium difficile, a bacteria that a lot of people have in their intestine, is the most common cause of this diarrhea. When other bacteria are wiped out from antibiotics, difficile can flourish and cause pseudomembranous colitis, inflammation of the colon. This is bad enough it could cause the bowel holes to form in the bowel and cause septic shock. Bad. So These infections are really bad because they're resistant to a lot of bacteria. They're hard to treat. New treatments include, there it is, fecal transplants to replace the healthy bacteria that will suppress the C. difficile. The residue remains in the large intestine 20 to 24 hours. No food breakdown occurring except what the bacteria are digesting. Vitamins made by the bacteria living in the intestine, water, and electrolytes are reabsorbed. The major functions of the large intestine are propelling the feces to the anus and defecation, getting rid of them, pooping them out. The large intestine is important for our comfort, but not essential for life. If the colon is removed, the ileum can be brought out through the abdominal wall in a procedure called an ileostomy. You'd wear a bag there to collect the feces outside the body. Motility in the large intestine. We have the haustra, the sacs, they can contract because remember, they have those three little bands of muscle, the tennae coli. The longitudinal tennae coli can contract, and these haustra will then contract in order sequentially to move things along. There might be some very slow segmentation, mostly in ascending or transverse colon. The gastrocolic reflex is initiated by the presence of food in the stomach. When food hits the stomach, you'll have movements of the intestines, slow, powerful peristaltic waves. They're activated 3-4 times a day to move the feces along. The descending colon, the sigmoid colon, those are storage reservoirs for feces that we're soon going to be removing from the body. If you have a low fiber diet, the colon, because you don't have enough stuff passing through, the colon can get narrow, and strong contractions could increase the pressure on the wall. That could have little pouches or herniations that pop out of the haustra. We call them diverticula. That by itself is not horrible, but diverticula can turn into diverticulitis where they get inflamed. Then they could rupture, leak feces into the peritoneal cavity. That could kill you. Also, the presence of the diverticula, half of people over age 70 have it. If you think about it, this is one of those things where you talk to the old people, have you been eating? Have you eaten enough today? Well, if they don't have enough food to push on, the walls are pushing on too hard, trying to find something to move along, and that's what causes the diverticula. Anyway, real common in the sigmoid colon. Let's move on. IBS, irritable bowel syndrome. This is a functional GI tract disorder. You can have abdominal pain again and again, stool changes, bloating, flatulence, nausea, depression. Stress is a real common factor in irritable bowel syndrome. Stress management is one of the most important forms of treatment for irritable bowel syndrome. Okay, defecation. This is pooping, getting rid of the feces. Mass movement supports the feces towards the rectum. Distension will initiate the spinal defecation reflex, and the parasympathetic will send signals stimulating contraction of the sigmoid colon and rectum, and the internal anal sphincter will relax. Conscious control allows relaxation of the anal sphincter. Remember, the anal sphincter is a skeletal muscle. We control it. So we decide whether or not we want to open that one up. So when we do, the muscles of the rectum will contract to force feces out. A lot of times this is helped by the Valsalva's maneuver. That is, take in a deep breath, hold your breath while you squeeze to poop. When you do that, you're squeezing the abdominal organs are pushing against the air. So we close the glottis, contract the diaphragm. The abdominal walls increase in pressure. The levitator anti-muscle contracts, lifting the anal canal upward, allowing the feces to leave the body. So there we have the how-to picture, but that's what we just talked about. Okay, so let's talk about diarrhea. Diarrhea, watery stools, are when the large intestine doesn't have sufficient time to absorb the remaining water. Causes: irritation of the colon by bacteria, jostling of digestive viscera. So marathon runners could be running so much that they can't use the intestine normally, and then they have diarrhea. Irritation by bacteria. So you eat something, it's got the wrong bacteria in it, you got food poisoning. The body could literally say, nope, I don't know what, we don't want that, get rid of it now. Do not take the time to reabsorb those nutrients, pass it out, get it out now. That's diarrhea. If you have prolonged diarrhea, you could lose so much water that you dehydrate and your electrolyte imbalance goes off. This could give you acidosis, loss of potassium. If you lost enough, that could kill you. So yes, people can die from diarrhea. There's a parasite named Giardia that can give people diarrhea severe enough to dehydrate and die. Anyway, constipation. When stuff stays in the colon for too long and we absorb too much water, the stool gets really hard, really difficult to pass. This can happen if we're not getting enough fiber. Fiber helps move things through. If we're not getting enough fluid, if we don't have proper bowel movement habits, if we're not using the toilet as often as we should, if we're abusing laxatives, or if we're not exercising enough, all of those can make it difficult to pass stools. Okay, part 3, physiology of digestion and absorption. Hold on a second. Okay, I just checked, and we're about halfway through this last section. They're on slide 63, and there are less than 120 slides, like 113 slides or so. Anyway, let's go through the physiology. We've talked anatomy and done a little bit beyond that, but now we're really going to hit physiology hard. So digestion breaks down ingested foods into chemical building blocks. Only those tiny little molecules are small enough to absorb across the wall of the intestine. Digestion is catabolic. This should remind you of cut. Think of scissors cutting. They are breaking big molecules into little pieces. And intrinsic and accessory gland enzymes are involved in digestion. Yeah, we've got that. Enzymes do hydrolysis. The name tells you what they do. Lyse means to break, hydro means water. You are taking, say, a carbohydrate that you want to break, and you shove, say you have a dimer of say you have starch. Okay, you got starch and you want to break off a glucose. You would shove a water up in there, and as you break the bond, you put an H on one side and an OH on the other. You take the water apart and you have broken off a glucose. That's hydrolysis. All right, so absorption is taking the things out of the lumen and putting them into the rest of the body, meaning into the blood, into the lymphatics, where you send it off to the liver for processing. Tight junctions mean that the molecules have to pass through the epithelial cells, not between them. This is good. We don't want parasites getting through easily. Materials enter the cell through the apical membrane, just like the word apex, that's on the side that's facing the lumen. They exit through the basolateral membrane. That is, the B should remind you of blood. That is next to the blood. The lipid molecules are absorbed passively through the membrane. Other ones are absorbed through active transport. What's the difference? Well, you remember membranes are made out of phospholipids, so lipids are going to go across really, really well. Other things don't cross phospholipids that well, so we have to actually find a method of pumping them across or helping them across. Most nutrients are absorbed into before the chyme gets to the ileum. So we go through the duodenum, we go through the jejunum, the first two parts of the small intestine. And at that point, we got most of the stuff out. All right, so the villi of the small intestine, remember they have microvilli, brush border on them. And for things to be absorbed, they have to go across the epithelial cell, and then they have to cross some interstitial fluid, get into a capillary. All right. Processing of nutrients. Only monosaccharides can be absorbed. We can't pull a starch across. We can't even pull a disaccharide. It's got to be a monosaccharide, a single sugar. So starches, disaccharides broken down into oligosaccharides, disaccharides. We start the breakdown of carbs in the mouth with salivary amylase. Then we're going to try to break down into lactose, maltose, sucrose. But the final breakdown is happening in the intestines, where you get down to glucose, fructose, galactose. The enzymes from the mouth, they get denatured when you hit the stomach. You can't do any of that in the stomach. All right, so we've got that. You understand that we start in the mouth, we pause the going through the stomach because we denatured everything. Then we hit the small intestine, and pancreatic amylase comes in, the brush border enzymes come in, and we're going to break everything down into monosaccharides like glucose, like fructose, which we absorb. All right, steps of the digestion of carbohydrate. What we just said, pancreatic amylase will break down the starch and glycogen that the salivary amylase didn't get. That's a typo. It's not amylase, it's amylase. The brush border enzymes, we have dextrinase, lactase, glucomylase, maltase, sucrase. We're going to break those down into lactose, maltose, sucrose, and then go even further to glucose, fructose, galactose. That is the plan. You have to break them down as small as possible to get across the epithelial cells. Monosaccharides co-transported across the apical membrane with absorptive epithelial cells, mostly through secondary active transport with sodium. Monosaccharides exit across the basolateral membrane through facilitated diffusion. So we're going to get them into the epithelial cell through active transport, and then we'll use facilitated diffusion to get them out of the epithelial cells into the blood. Here is a nice drawing showing us what's happening. So we've broken our glucoses down here into our lovely blue glucoses, and we have sodium. We are transporting both in the same direction. This is going to be, so as we do that, we then have our monosaccharides that are coming in. The sodium is pumped out through using ATP, and we have a monosaccharide carrier molecule that will move the monosaccharides across the basolateral layer so they're next to the capillaries and ready to go in. Okay, and no, I'm not going through each one of those individually. We just talked about it. All right, people who are lactose intolerant don't make that much lactase. That, by the way, is the normal condition. A lot of people in the West act like that is abnormal. No, most of the people in this world are lactose intolerant. In fact, geneticists, maybe not the anatomists in this book, but geneticists talk about people who are not lactose intolerant as being lactase persistent. See, we all, as babies, when we're drinking mama's milk, make lactase. The people who are tolerant to lactose just make lactase their whole lives. The people who are not stop making lactase, just like all the other mammal babies do. Um, anyway, so if you don't have lactase, any lactose that you eat stays in the intestines undigested. It makes an osmotic gradient, so you don't absorb water, and it gives you diarrhea. It could also pull water out of the interstitial space into the lumen, again giving you diarrhea. The bacterial metabolism of the stuff you didn't digest could make gas. So on top of the diarrhea, then you have bloating, you have flatulence, you have cramping. Treatment. You put lactase enzyme drops in the milk, so when you drink it, you have the enzymes too. Or you take a lactase tablet before you do milk products. Cheese, ice cream, whatever. Digestion of proteins. Protein that we get is not only dietary, but also digestive enzymes and proteins for breaking down mucosal cells. But most of it's stuff we have to eat. Proteins are broken into big peptides, large polypeptides, small polypeptides, and finally down to individual amino acids. With a few, well, I shouldn't say that, mostly individual amino acids, amino acid monomers, but also a few dipeptides, meaning 2 amino acids hooked together, or tripeptides, 3. Digestion of the protein starts in the stomach. When pepsinogen hits the acid, it turns into pepsin. And so that enzyme, pepsin, is good at digesting proteins as long as we're in the low pH of the stomach. The minute we pass the chyme into the duodenum, we denature the pepsin. That means that we have to, in the small intestine, use trypsin or chymotrypsin or carboxypeptidase. We use other enzymes because we denatured our pepsin. All right. And at the brush border, we finish breaking things down even further, and we absorb mostly single amino acids, but also some dipeptides, some tripeptides. All right. Steps of protein digestion in the intestine. Just what we saw in that picture. The pancreatic proteases, trypsin, chymotrypsin, they're going to break proteins even smaller. Carboxypeptidase will take 1 amino acid at a time off the end. The brush border enzymes, we have aminopeptidase, carboxypeptidase, dipeptidase. They'll break off, they'll take individual amino acids out, or I should say, they will take small peptide chains and break them into amino acids. Amino acids are co-transported across the lumen side of the epithelial cell using secondary active transport, either sodium or hydrogen pumping. The amino acids will exit across the basolateral down by the blood with facilitated diffusion. So this should look pretty dang similar to what we saw with the carbs. All right, and it does. All right. Digestion of lipids. This will be a little different. Emulsification. The triglycerides and their breakdown products, they don't dissolve in water. You need to mix them with bile salts that break the large fat globules into small ones. Why? That increases the surface area. That gives more surface to have enzymes working on. The digestion. Pancreatic lipases will break fats down into monoglycerides, and 2 free fatty acids will form little circles, little bubbles of fats and fatty acids we call micelles at that point. These are coated with bile salts and lectin. Lipid products will leave the micelles and cross the epithelial membrane through diffusion. Now remember, that's different. The proteins needed an active transport, the carbs needed an active transport. The lipids do not. Why? because the membranes they're crossing are phospholipids. The chylomicrons are formed, lipids, products converted back into triglycerides. Those are what we commonly call fats, packaged with lecithin and lipoproteins forming a chylomicron. Chylomicrons cross the blood side, the basolateral side, with exocytosis and enter the lymphatic lacteals. This is why the lacteals are called lacteals, because lact, like lactose, like milk, that is lymph that looks milky white from all the little chylomicron drops in it. And eventually that's going to dump into the venous blood at the thoracic duct, which means it'll have the long route to go before it gets to the liver, but it'll eventually get to the liver where we work on it. Once we're in the blood, the chylomicrons are broken into free fatty acids and glycerol by lipoprotein lipase, so they can be used by cells. These short chain fatty acids can go directly into the blood. All right. Again, we've got that. Straightforward. All right, digestion of nucleic acids. I want to say something before we continue. There are 4 major biological molecules, and I'm talking about proteins, carbs, lipids, and nucleic acids. So we've gone through the other 3, and all of those we were using for as nutrients. They're also much more energy rich. Lipids have the most energy per gram. Carbs would have the next highest. Proteins would have the next highest. Then you get to the nucleic acids. They might be useful. We might use, we might eat a cell and you break down its DNA to use to build your own DNA, but you're not going to get much energy out. So most books I've seen, they don't describe this as a nutrient. So any food you eat that has cells has DNA and RNA in it. You've got nuclei. There are pancreatic enzymes called nucleases. They're going to break those down into nucleotide monomers. I'm talking about the A's and C's and T's and G's, the adenine, cytosine, guanine, thymine. Yeah. Those brush border enzymes, nucleosidases, phosphatases, they break the nucleotides down further into free nitrogen bases, pentose sugars, phosphate ions. Those things can be used to make your own. The breakdown products are actively transported by special carriers in the villi on the epithelium. So there we have pictures. They're talking about absorption of vitamins, electrolytes, and water. In the small intestine, the fat-soluble ones, the A, D, E, and K, are carried by little micelles of fat and diffuse into the absorptive cells. The water-soluble vitamins, the C and D, are absorbed by diffusion or by passive or active transport, either one. B, that's large, it's charged, it doesn't cross membranes well, but it binds with intrinsic factor and then it's absorbed through endocytosis. Very active transport. The cells reach out and say, oh, thank you, and pull that inside. In the large intestine, vitamin B, vitamin K are absorbed from bacterial metabolism. So our bacteria in our microbiome, bacteria are going to make some vitamin K, some vitamin B, and we will absorb and use those. Okay, absorption of electrolytes. Most ions are transported actively along the small intestine. What that means is we don't want to pee out things that are precious. We don't want to poop out things that are precious. We try to keep them. Iron and calcium absorbed in the duodenum. Sodium is used for coupling, meaning all the things that we saw before, like how you absorb carbs, how you absorb proteins. This kind of stuff, we need sodium. See the sodium right there? All right. So we're going to absorb those all along the chain. Where was I? all along active absorption of glucose and amino acids. Calcium, active transport, what did I say calcium? That's chlorine. Actively transported chlorine. Potassium diffuses in response to gradients. We lose the potassium if we're not good at absorbing water. And usually what we have left in the intestine, the electrolytes we're going to absorb. Iron and calcium absorption is related to how much we need. Iron absorption is, we store that in the mucosal cells in ferritin. When it's needed, we dump it in the blood as transferritin. Calcium is absorbed with vitamin D. If you don't have vitamin D, you can't absorb calcium well. If someone has, say, osteoporosis, they are probably going to be not just told to take calcium supplements, but told to take vitamin D so they can absorb it. Also, affecting calcium absorption is parathyroid hormone. If you remember, we talked about this before when we did the skeletal system in AP1, but if you have too much parathyroid hormone, you are not going to reabsorb calcium very well. Sorry, you're not going to put calcium into your bone very well. So parathyroid hormone is going to say, hey, we need calcium in the blood. So yes, it would in this case absorb calcium, but if you have too much, say you have a tumor that causes too much parathyroid hormone, it's also going to cause you to break down bone to free up the calcium and put it in the blood. Okay, so absorption of water. We absorb 9 liters of water, most from the GI tract secretions that get into the small intestines. 95% of this we absorb in the small intestine through osmosis. Almost all the rest of it is absorbed in the large intestine. Net osmosis occurs if the concentration gradient is established by active transport. Remember, water wants to dilute things. Water uptake is coupled with solute uptake. If you don't get enough solutes, you're not going to be able to move the water. A malabsorption is when you have trouble with absorption. Malabsorption can be caused by anything that interferes with delivering bile or delivering pancreatic juice. If you have damaged intestinal mucosa from antibiotics or from hostile bacteria, that could cause a problem. You also, there is a problem with gluten for people who have allergies. Gluten-sensitive enteropathy, pathy for disease, entero for the inside, the GI tract. Another name for this is celiac disease. It's a common malabsorption disease. The immune system has a reaction to gluten. Sorry, if the immune system has a reaction to gluten, the gluten will cause the immune cell will cause immune cells to attack the intestinal villi on the brush border. What do you do to fix it? You have less gluten in your diet. You try to eliminate the gluten. Let's talk about making the GI tract now. The primitive gut, meaning what you have as an embryo. The epithelial lining is formed from endoderm, the inner layer. The rest of the GI tract is formed from mesoderm, the middle of the 3 layers that you first get. The oral membrane that becomes the mouth, that's formed when the foregut of the primitive gut fuses with the stomodeum. Now, I want to point out that humans are deuterostomes. That means we form our butt before we form our mouth. Anyway, the first opening will become the anus, and the second one will become the mouth. I don't really care about proctoduum. I'm not going to worry about that. 5 weeks. Alimentary canal. Yeah, so it takes 5 weeks for the alimentary canal to become a continuous tube from mouth to anus. Shortly after that, accessory organs will bud off of the mucosa. Great. Here we have the developing embryo. Homeostatic imbalance. A cleft palate or a cleft lip happen when the palatine bones and the palatine processes of the maxillary bones, one or other or both, don't fuse properly. This can be a problem because it'll be difficult for the child to suck properly, and that means they can't nurse well. Tracheoesophageal fistulas are when you have a hole, a fistula, between the esophagus and the trachea. This is really, really bad because you could get food or liquid down into the lungs, and that you don't want that. Cystic fibrosis is a genetic disease that makes thick mucus that can block the pancreatic duct. If that happens, you can't put pancreatic fluid in. That means that chyme will be hurting the intestine. It means you don't get all the enzymes where into the organ where most of the digestion occurs. It's bad news. Okay. So the digestive system after birth, the fetal nutrition occurs in the placenta, but the GI tract is stimulated to mature by amniotic fluid that the developing baby will swallow in utero. The newborn's rooting complex, or rooting reflex, sorry, helps it to find the nipple. Sucking reflex aids in swallowing. A newborn human baby will double in birth in 6 months, and it'll be capable of having adult food by 2 years. I know a lot of people who don't treat kids that way. Oh, she can only eat chicken nuggets. No, that's crap. They can eat a full adult diet. Gastroenteritis, inflammation of the GI tract, that's a problem. And cholecystitis, ulcers that can become a problem in middle age. Aging in the GI tract. The GI tract activity declines with age. It makes less digestive juice as you get older. The absorption gets less efficient. The peristalsis, the movement through the GI tract gets slower, and that has a lot to do with why people don't have as bowel movements as often. Also, not eating enough. That's again, they need more fiber. They need to eat more. That's how you keep the bowel moving. As you get older, your hold on. No, that's what I was trying to avoid. Hold on. Okay, one of the other things. As you get older, your sense of taste, your sense of smell are going to become weaker. That means that you might find old people making their food super salty because they can't taste the salt anymore. They might have periodontal disease, problems. That's why a lot of old people lose their teeth. Diverticulosis, where you form the diverticula, the little pouches coming off the haustra. Fecal incontinence, difficulty. Why do people wear Depends diapers? They're having trouble keeping their their feces in. Cancer of the GI tract is a pretty common one as well. All right. Stomach and colon cancers don't usually give early signs or symptoms. If they metastasize, they frequently cause liver cancer. Oral cancers can be detected during dental exams. Most of the GI cancers are treatable if you catch them early enough. And how do you prevent them? Regular dental and medical exams. That is the end. We are done. Let's shut this down.